The IDEXX Enterolert-DW test detects Enterokokki in drinking water in 24 hours with no confirmation step. Enterolert-DW is available in a 100 milliliter format and can be used only for quantification. In the next few minutes, you'll learn how to use the Enterolert-DW test with a Quanti-Tray system to determine the number of Enterokokki in a water sample. IDEXX offers two sizes of Quanti-Tray. Both work with the Quanti-Tray sealer. The standard Quanti-Tray is a disposable 51-well tray that provides counts up to 200 per 100 ml sample without dilutions. The Quanti-Tray 2000 is a disposable 97-well tray that provides counts up to 2,419 per 100 ml sample without dilutions. You will need the Enterolert-DW reagent, a sterile, transparent 100 ml vessel, a temperature-controlled incubator, a Quanti-Tray or Quanti-Tray 2000, Quanti-Tray rubber inserts, a Quanti-Tray sealer, and the included most probable number table or the IDEXX automated MPN generator program found at IDEXX.com. All of these items are included in the Quanti-Tray system and are available from IDEXX. Turn on the sealer and then wait until a green bar appears on the display. The sealer has now reached the appropriate sealing temperature. Separate one reagent pack from its strip, taking care not to accidentally open the other packs. Snap open the reagent pack and pour the powder into a 100 ml water sample. Cap the vessel and shake to dissolve the Enterolert-DW powder. Shake until the reagent dissolves completely. Use only a permanent felt tip marker to label the Quanti-Tray. Adhesive labels may adhere to the sealer roller, and ballpoint pens may puncture the paper. Hold the Quanti-Tray at the top with the well side facing your palm. Push the top edge of the Quanti-Tray in with your free hand while squeezing the tray into a circle. Open the Quanti-Tray by gently pulling the foil tab away from the plastic side. Be careful not to tear the tab. Do not touch the inside of the tray. Pour the sample mixture into the Quanti-Tray, avoiding contact with the foil tab. Place the filled Quanti-Tray on the rubber insert, making sure each well fits its corresponding hole. Push the insert with the tray into the sealer until the sealer grabs the tray and pulls it into the slot. If you need to reverse the motor, press and hold the reverse button. However, do not reverse the motor if the rubber insert is completely inside the sealer. The sealer distributes the sample mixture into the Quanti-Tray wells, seals the wells, and partially ejects the sealed tray. Remove the rubber insert and tray from the sealer. Incubate the sealed tray for 24 hours at 41 degrees centigrade, plus or minus 0.5 degrees centigrade, or according to your local regulations. After incubation, mark each green well with a permanent marker, including the large overflow well. Count the marked wells and then refer to the MPN table included with the Quanti-Trays. The most probable number for this example is 20.7 per 100 ml. If using Quanti-Tray 2000, count the overflow well as a large well. Any color change from blue to green means a confirmed positive for Enterokokki. Here is an example with both strong and weak color change. There are a total of 17 positive wells. If you are unsure whether a result is positive, compare the sample side by side with a known negative sample. If the results are ambiguous based on the initial reading, incubate up to an additional 4 hours, but not to exceed 28 hours total, to allow the color to intensify. IDEXX recommends that you run a quality control procedure for each lot of Enterolert-DW tests. You will find the quality control procedure in the Enterolert-DW product insert. Remember, incubate samples for 24 hours or up to 28 hours total. Samples with no green color are negative for Enterokokki, and samples with any change to a green color are positive for Enterokokki. Always follow these testing guidelines. Use only sterile, non-buffered, oxidant-free water for dilutions. Store tests at 2 to 25 degrees centigrade. And always follow aseptic technique. Because these procedures may not reflect your local regulations, for compliance testing, be sure to follow all local regulatory procedures. Keep these tips in mind. No confirmation steps are needed. Positives before 24 hours and negatives after 28 hours are also valid. Up to 10 trays can be stacked for incubation. Do not place trays in a box or a bag. Dispose of all samples and materials in accordance with local regulatory guidelines. If you have questions or need more information, call the IDEXX water microbiology experts at the number on your screen.